Hello, today we are going to talk about enzymes. Enzymes are extremely important biological systems. An enzyme is a molecule that speeds up a chemical reaction by bringing the substrates together in the right orientation. Here is our enzyme. We know that this right here is the active site because the substrate fits inside it perfectly. And the active site is where the interaction happens between the substrate and the enzyme. So here's also another possible substrate. You see how the charges oppose? And these ones do not? Keep in mind that enzymes are very specific. They only catalyze very particular reactions. Every single enzyme catalyzes one reaction. Before we move on, let's discuss anabolic and catabolic reactions. In anabolic reactions, we basically have smaller pieces coming together to build larger molecules. In catabolic reactions, we have larger molecules being broken apart into smaller pieces. Enzymes play a role in both types of reactions. Let's begin with catabolic. Here's a molecule. So in a catabolic reaction, we want to start with this larger piece, and we're going to break it apart to end with two smaller pieces. In an enzyme, it's going to enter the active site as one piece. Then the enzyme is going to break it apart and release it as two. A real-life example would be lactose digestion. Pretend this is lactose. It's going to enter the enzyme. It's going to break it apart. Now we have galactose and glucose. Now let's discuss anabolism. In anabolic reactions, we begin with two smaller pieces rather than one large piece, and we want to end with a larger piece. The way this is going to work is that the two pieces are going to enter the active site separately. So this is going to move in, and then this one's going to move in. And then they're going to be put together by the enzyme and released as one molecule. This is really, really important because in a solution without the enzyme, it's very unlikely that these are going to be floating around, bumping into each other, until finally they come into each other just the right orientation to become that one piece that we want. The enzyme orients them in the way to induce that fit, to stop them from wiggling around so much. One real-life example that you could think of is ATP production. Pretend this is the phosphate and this is the ADP. And this is the ATP synthase. The phosphate enters, then the ADP, and it comes out as ATP. Of course, it's not really how it works because we don't have a membrane, but just pretend. Okay. So we need to control enzymes. We can't just let them work all the time. Or they need to be regulated. One way we do that is by competitive inhibitors. Here's our competitive inhibitor. The reason why we call it competitive is because it's shaped similarly to the substrate. See how it's a little similar? It's not exactly the same, but it can actually fit inside the active site. And when it does fit inside the active site, the substrate can no longer get in. So that's how competitive inhibitors work. They don't bind covalently, so they are eventually released. Another type of inhibitor is allosteric. An allosteric, or also called non-competitive, works by binding somewhere other than the active site, or right here. And what happens is when it binds to this site right here, it changes the shape of the active site so that the substrate can no longer fit inside the active site. Watch it. Now the substrate can't fit anymore, and since it can't fit in the active site, it can't be put together. Let's try it with this one. This can't get inside anymore, so these can't be put together. Or our lactose. The lactose can't get inside. Unlike competitive inhibitors, these actually do bind covalently. Now going back to our anabolic and catabolic, just remember, these models aren't usually perfect. Enzymes are specific, like I said earlier, so this would not catalyze this reaction and this reaction. It would only catalyze one or the other. We're just using it as an example for both. So if it catalyzed this, it would not also catalyze that. So yeah, that's how enzymes work.